So today I'm talking about my postdoc studies, one of my postdoc studies, Turacos. So yeah, I finished my PhD. I just try to give a little bit information about me. So I finished my PhD 2008 in Turkey. Then I applied a postdoc studies, and yeah, I picked up the MNH to made the application, and I made my application over there, and they evaluated, and I started 2010 as a postdoc over there and working on actually the planning work was about woodpeckers. So we did some previous studies about green woodpecker. We published, and then I applied my study, my proposal. It was about African woodpeckers, Campatera group. So anyway, they evaluated, and I got the acceptance letter, and I moved there in New York. And I thought I was going to start working on woodpeckers. So I looked at collection, I tried to make my arrangement, but at that time, we learned somebody from California already finished the woodpeckers in Africa. So it was about kind of, they were preparing to submission process or something, anyway. And then it was really big disappointment for me, because I applied my project and everything is just evaluated, and I thought I was really prepared myself, to working on woodpeckers. Then I'm working with George Biocultural over there. He's right now emeritus curator in American Museum. And then he said, okay, we should just pick up another group. We have to be very careful and try to find another group. And then we looked at the collection. Then I picked up the Turacos because it's very, interesting bird group, endemic to sub-Saharan Africa. Nobody really worked on Turacos. There are a couple of different studies published. One of them, Morrow, published in 1960s, a very big monograph based on the morphology in Aves. Then, I think end of 1900s, 1999, I think, somebody published based on the captive birds, the phylogeny. They published the phylogeny based on the captive birds, but nobody actually published really, real population to try to understand the real population structure. And I checked the collection. I found very broad range of the sampling about Turacos in American Museum, and also I checked the Field Museum collection, I checked the Yale Peabody Museum collection, and we can easily reach the samples. Then we started arranging the project. Then after that we thought we could finish within 2 years, but it took 10 years to finish because project got bigger, and bigger. Then I got these things, and we published, I said a big monograph, but it's a very comprehensive monograph in 2020. So we decided to continue with the go-away birds, and at that time the COVID-19 came, and we all locked down, and I just stuck in Turkey. I couldn't go to New York, and then I thought we are going to finish the study, but fortunately last year we decided to continue the study, and we moved to next generation sequencing. Now we are exploring the new stuff about go-away birds. There are also some hidden genetic diversity within gray go-away birds. I'm sure the other go-away birds also has some hidden genetic diversity and nobody knows. So it's very interesting. So anyway, this is my short story. So I can right now move to the presentation. Today, I'm talking about Turacos, and I'm going to introduce to some scope of phylogeographic research on Turacos because I'm interested in phylogeography. The phylogeography means, probably you all know, but I just try to give some information. Phylogeography interests the genetic variation of the geographic variation of the genetic diversity within species or within closely related species, not between the species. So we first focused on the phylogeography of Turacos, and we picked up every species within Turacos and tried to understand the genetic variation pattern within each species in green Turacos. Then try to understand the population limits and species limits. Then we move to phylogenetic structure of the group. And yeah, this is all, I made in my study. So here the little definition of the phylogeography, study of the historical processes that may be responsible for the contemporary geographic distribution of individuals. And phylogeography helps explain the patterns of speciation and adaptation. And we use the DNA sequencing. Actually, we can use morphological characters to get the phylogenetic tree to understand the phylogeographic pattern maybe, but the genetic markers also very important to understand the population limits, species limits, and genetic, variation of the genetic diversity. This is my publication. It came from American Museum Novitates and 2020. So it includes all green Turacos. Green Turacos has traditionally recognized 18 different species. So they have some special pigment, which is called turacin and turacoverdin. So also, well, I said the traditionally defined species, the 18 different species. When we get the DNA results, it turned out 25 different species. And there are some hidden genetic diversity. I'm going to show you all of these. Not all of them, but I picked up some special examples, and you can understand what kind of genetic diversity that we have. And in the small scale and the broad range, we have some interesting genetic pattern. And we published in American Museum Novitates all these results. So we focused on the DNA. So I put here, we are right now in the genomic age. So there is no excuse to get the complete genome from the specimens, from the bird species, because, right now it is really, not really cheap, but it's cheaper than before, and there is no excuse. But at that time, I started 2010, and that time, there was no next generation sequencing stuff. I think next generation sequencing introduced in the phylogeography, in ornithology, or in biology in 2010 or 11. So it was at that time very, very, very expensive, and we didn't have any, and I didn't have any experience at that time. Anyway, we focused on the first individual genes to understand the genetic variation. We focused on the individual genes because I also focused on the museum specimens. The museum specimens is very old and travels a lot, and lots of people touch these museum specimens. This is the contamination problem also. And I try to cut the toe pads from each species, each individual, and clean these toe pads very carefully, and then digest them using some special chemicals and get the DNA. After getting DNA, we need to get the gene, some specific gene, to understand the variation pattern. But there are lots of genes in the genome, so that we first focused on the mitochondrial DNA, because mitochondrial DNA always evolves faster than nuclear DNA, other groups, other gene groups. So it is really useful to get the mitochondrial DNA to understand the recent history of the species and some diversification pattern. So we focused on the mitochondrial DNA, and we picked up the ND2 gene. So ND2 gene is protein coding gene and has lots of variation in birds. So it's a very useful gene. So we picked up this gene, but this is 1041 base pairs, and when you focused on the old museum specimens, like 100 years old, 120 years old, maybe 80 years old, maybe 50 years old, sometimes it is not possible to get the complete gene using one experiment, one primer set. So we have to design different primers, like here, 1, 2, 3, 4, 5, I made the numbers. So, and then we need to get the short piece of the gene, like a 100 base pairs, and then we need to get some overlap using the different primers to understand is there any contamination or not. So different primer sets always try to catch the same DNA sequence. So if they get the same DNA sequence, we can trust the data, so we can understand there is no contamination. So because of the situation for one individual, I set up 10 different, sometimes more than 10 different experiments. So from one individual to get the complete DNA, complete gene, so it took really, time. So that's why, this study took really time, and sometimes I stayed, in the museum. There, in my life, there was sometimes the museum at the night at the museum, like a movie. So, try to get all these sequencing using traditional techniques. So anyway, it took time to get this data ready. After that, we moved to some other markers using the nuclear markers. Nuclear markers, always evolves slower than mitochondrial DNA, and also mitochondrial DNA inherited from the females, not from the males. So when we get some genetic diversity from mitochondrial DNA, we always understand the female history, not the male history, but we need to understand the male history also to discuss the gene flow between populations and to evaluate the taxonomic situation, taxonomic problems. So anyway, first we focused on the mitochondrial DNA, and then we moved to some nuclear DNA after that. But mitochondrial DNA results looks very interesting. So the mitochondrial DNA shows us really, big, lots of genetic diversity. So I try to put this genetic diversity here in this slide. You all can see the different colors and different, circles has different diameters. So each different color indicates a different DNA sequence. So let's say each color has different DNA. And then between the colors, all these, pies, you can easily see the small dots. And when you count these small dots, you can understand the difference, because all these small dots shows us the mutation numbers. For example, you can look at the 600 base pairs between two different populations. When you get the 600 base pairs, and you can just count these small black dots, you can easily understand how many base pairs different between them. For example, some groups has 30 different base pairs, some groups has 10 different base pairs, and you can focus on this group. You can easily see some groups has only one base pair difference between different DNA sequences. So this actually gives us some basic information how many genetic diversity within these Turacos. So we got this analysis, and nobody actually knows how many genetic divergence and differences between these groups, because they're all green. They all look like similar. Based on morphology, you can expect some differences, but some differences really, huge, because in vertebrates, not only the birds, the other mammals, if you want to get one mutation or 2% mutation, you need to wait 1 million years. So this is quite a history here when you think about the Turacos, because there are lots of mutations. Let's say the 1 million years passed, and then within 100 base pairs, only 2 different base pairs can change. I hope I could explain. And I just focused on these different groups here. the one group here, this is the one species group. Another species, Bannermani, from Cameroon. So Leucolophus, Erythrolophus from Angola, and Macrorhyncha and Verauxi from west part of Africa. They look like based on the morphology, general morphology, they look like the same. But when you focus here, and you can count all these dots, there is a big, big, big split. So they are monophyletic, they are coming from the one common ancestor, but I mean, they really diversified, they really diverged. And I mean, very long time ago. And we got this information, and we got this information, and after that we put this information on the map and try to understand the geographic structure of the variation using some geographical barriers. For example, this is Porphyriolophus. Porphyriolophus, that species, distributes in that area, the green area. And this species has two different subspecies. One subspecies has brownish breast. Another subspecies, you can't see the brownish breast. So the one subspecies distributes in the north and one subspecies distributes in the south. And we got these samplings, all these are sampling points, and we got two different DNA group. Let's say the two different DNA group. One group has black, another group has white. When you focused on the black group, they concentrate on the north, and then white in the south, but there is also the black occurs in the south, but not many. So this line indicates the Zambezi River Valley. This is the geographical barrier between two different subspecies. Recognizable barrier. And when you look at the map, you can easily see this valley. It's a big valley and a big river. So this big river makes them two different subspecies, but still some genetic flow historically, probably they got some historical genetic flow, and that's why that black dots occurs here. But when you look at the morphology, you can see the brownish breast consistently in the south. So we recognize, based on all this information, we recognize this group has two different phylogenetic species. I mean, we all use the biological species concept, but two different species has different kind of history, and we put the history in the species concept, we can talk about the phylospecies concept after that. So we recognize two different species, and then each subspecies deserves to be one species, we believe, based on these results. And some other Turacos, for example, one of them, yellow-billed Turaco, here. And it distributes in the western part of Africa. And again, this species has two different subspecies. One subspecies distributes in very west part of Africa. One subspecies distributes in the west part of Africa, here in the middle, close to the middle. So between them, I mean, I can use. Between two different distributions, there is a one blank area which is called the Dahomey gap. This is the savanna area. This is the rainforest species. And we try to get the DNA again, and then we got the DNA, and based on the DNA results, they are monophyletic, and one group occurs here and another group occurs in a different color, and there is no gene flow between them. There is no sharing DNA sequence between them. So the Dahomey gap, the blank area, made a really geographical barrier between these two subspecies. After these examples, I'm going to show you the big phylogenetic tree. You can easily see all these relationships between different Turacos. And we also recognize two different species in this group, and because one one subspecies has red crest, one subspecies has black crest. They are consistent and morphologically consistent. But otherwise, when you look at this bird, this is also Turacos, but different color. Very nice bird, and has very wide separate distribution range. One of them, rosy, distributes in the middle of the Africa. Another one, west part of Africa. So in the same kind of range, when we look at the Macrorhyncha, we found some substantial amount of genetic diversity between these two groups. But the same kind of range Violacea has, and we couldn't find, within Violacea, any differentiation. So it means probably they just got this distribution range. There are some refugee hypotheses for Africa. They got this distribution range probably very fast, and they didn't get enough time to differentiate it. So another one also the same. So there is no really big difference between the haplotypes, between the DNA sequences, but they have the big range. So this is kind of the different example. So also there are some surprising genetic isolation. One of them, Turacos schuettai, black-billed Turacos. So that species, very handsome species. You can easily recognize the difference. Based on the traditional taxonomy, people recognize two different subspecies. One of them greenback, one of them purpleback. It's very recognizable. There is no question about this. So one of them Schuettai emini, another one Turacos schuettai nominate subspecies, and it distributes in the middle area, the Congo area. And now we got these haplotypes, and look at these haplotypes. This is very interesting. Please don't forget this example. So there's a big difference. There's a big difference, huge difference. Okay, we found it, and we try to understand what is the barrier between these two subspecies. So we couldn't see any geographical barrier between them. There is a Congo River there, but Congo River didn't make any difference between these two subspecies, between these two distributions. So we couldn't really understand, and sampling size is not very common for the middle of the Congo. So we just leave it alone. We found these results, and we said there is a huge difference. But please don't forget this example. I'm going to come back. And another one, Turacos persa, that species. This is another green Turacos. There are two different subspecies here. When you look at one of them, you can see just below the eye, so there is a black line, another one, the white line. So there is only the difference between them. It's a small difference. I think the sexual selection is really worked for them. And then it has the continuous distribution in the west part of Africa. The Homie gap didn't make any geographical barrier for this species. But we found, again, between one of them Buffoni, one of them Perse, we found again two different genetic population structure within this group. And now we thought only this black line very important for the sexual selection for this species. And this is also another interesting example. And then we can move to some small-scale distribution. Example, this is Turacos hartlobi. This species only occurs in Kenya and environs of Kenya, Uganda, and north of Kenya, and north of Tanzania. But this species only use the high elevation habitats, mountain tops. And there is no morphological variation between populations. When you get the specimens from Usambara, and when you get the specimens from Nyuru, north of Kenya, there is no morphological differences. No. And there is no morphological differences between male and females also. They are all the same. And we got the DNA, and we found Usambara has own genetic structure, Kilimanjaro has own genetic structure, Algon has own genetic structure, and Algon monophyletic to other groups. And there are some gene flow in Kenya. There are some high elevation bird populations in Kenya. There are some gene flow between them. But all these species, I mean, all these populations, unique genetic diversity. And very small scale, only the Kenya. So we looked for the Congo. We couldn't find any genetic divergence between population, for example, Violacea, the purple one, purple Turacos. But this one, we found, I mean, substantial amount of genetic diversity within a very small area. And we tried to get some song information. We got some morphological measurements. We couldn't find any differences. We tried to get some song information. We couldn't. So we couldn't go there and make some recordings. But we believe this is kind of the cryptic species complex. Somebody needs to focus on this species and needs to work the behavior and mating system, maybe song, some other things. There is really something really hidden in this species group. And it's very important for the biodiversity conservation because in this age, we have, I mean, in the biodiversity crisis, we are losing the species even without describing them. Loss of genetic diversity, I mean, lots of different things we are losing because of the habitat destruction, climate change, global warming, etc., etc. So this is the, I mean, interesting example. Okay. So we did for each species, we did for this kind of study for each Turacos species, and we got all this information, and we established our unique lineage, unique genetic structure, and then we got mitochondrial DNA. Using mitochondrial DNA, we established all these unique structure, unique lineages. Then we got some introns, different markers, evolving slowly, and made some big phylogeny, and we got this phylogeny. And this ND2, this one shows us the mitochondrial DNA phylogeny. This one shows us the intron, I mean nuclear DNA phylogeny. There are some discordance between the lineages, but mostly, I mean, they look like the same. The resolution is a little low. I'm not going to give the more, I mean, methodological details. So after that, we also get with another slow markers, reg1, another 2,800 base pairs, but in total we got from the genome almost more than 4,000 base pairs. Then we got this species tree. And please, just please remember the Shuetai, the green back and the purple back, middle of the Congo. There is two different subspecies. I mean, this group, not sister. The one subspecies closely related to other Turacos, another subspecies occurs here. They are not even, I mean, they are not even diversified from the one common ancestor. Completely different. So it is very, very interesting point. And another one, Livingstone, I didn't show the results here, but also it has the same situation. There are lots of hidden genetic diversity in Turacos that we found. Then after that, we, I mean, suggested some new taxonomy, and it was 18 different green Turacos, and we evaluated some subspecies of species, and it is right now the 25 different species we suggested. And BirdLife, I think, accepted some of them. I checked the recent list. And also we changed some generic names. So based on this classification, we only just suggest in our paper, but it's kind of very interesting group. And at the end, our study integrates the two different DNA markers to refine the phylogenetic relationships within Turacos, confirming the distinct lineages that I try to show. And we have identified the significant genetic diversity and structure within Turacos, and unexpected genetic breaks in Turacos also we found. So I recently read something very interesting. We couldn't find really exact geographical barrier between some species, some subspecies. So this bird group has tyrosine, the spatial color pigment, and this color pigment has copper element. And recently I read something, and all these birds really follow the copper mines. Their distribution really match the copper mines. And I just try to get some information from Africa, all these copper mines, and put the map using the geographical information system and put this distribution, try to overlap to understand, is there any logical match between them? But I recently read, I found this information very interesting. So, I mean, this is general outline of the study, and in summary, the results indicate that there is a substantial amount of previously unrecognized genetic diversity in African birds, particularly in montane and lowland forest areas. And these results indicate that we need to re-evaluate the existing Avian biogeographic zones and historical distribution models for Africa. So we believe it's going to help for some African ornithological scenarios, biogeographical scenarios. And also Turacos, also very important in the culture and aviculture. So their red pigment, also tyrosine, has inspired myths-like stories of bleeding wings in the rain. And in some cultures, Turacos symbolize vitality and courage, adding depth their value in the aviculture. So some local people, some indigenous people, using these colorful Turaco feathers commonly found on their ceremonial costumes of the forest people of Central Africa. So it's kind of the, indication of the power, I think. Historically, it's very important. I just try to put this information because, I'm here in United Kingdom. I am working in University of Oxford with Andy Gosler, Douglas mentioned. So we just try to do something about ethno-ornithology, kind of the different perspective. During the lockdown process, so we all locked down, I just tried to make backyard for myself, in a home. So I decided to make some historical database using some historical literature. So this is why I am here right now, and this is why I'm visiting the museum collection. So I'm developing a historical database of African bird records and try to understand some ecological impact and cultural impact, because I'm getting the historical data like between end of the 1800s till 1950s and make maps. So I'm going to show you very briefly, next slide. And then try to understand the distribution patterns, and using some citizen science data like eBird, downloaded every observational records for every African passerine birds, try to make a match between them, try to understand is there any shrinkage or expansion pattern, and try to understand what's happening based on the climate change and also human impact, habitat change. We can use that. There are lots of online databases available for these kind of analysis. And my other question, assessing the change in distribution and plus cultural name loss over the time. So in Africa, there are different languages, and indigos are really interested in this ethno-ornithological perspective, and we try to get each species how many names they have got, and based on the climate change and the global warming, we know some species is going to be extinct or vulnerable or endangered when we get to 2100 or 2080. So how many bird names is going to be extinct in the local languages? I try to understand, I mean, in this study. So for this, I focused on this book, An Atlas of Speciation African Passerine Birds. So Mrs. Hall and Robert Morrow and David Snow made 2 volumes, really nice study, published in 1970s. I mean, they got all museum specimens. I think they used the Tring specimens. Also, they used some other museum specimens, 4 different museums, one of them Durham museums. Made a really nice maps and tried to discuss the speciation pattern. But they didn't give any latitude, longitude information for this distribution. Well, they put the dots on the maps, but they didn't give any latitude, longitude information. So I put Andy's picture here, Andy Gosler. So this is one of the map. I digitize this map, make a georeferencing, and converting to this map like this map, put all the dots, and try to extract the latitude, longitude information using geographical information system, and extract this data and make a historical database. So, I mean, we can use it, and I'm just trying to use it. I just finished for the bulbous, and also all these larks, and I think I digitized more than 20,000 dots. Right now, I have the data. So also at the same time, I'm downloading the data from eBird for this species. I also try to evaluate the taxonomic situation because some species has different taxonomy right now, and make a match, make a overlap maps, and try to match and try to understand the shrinkage and expansion pattern and put some ethno-ornithological perspective on these maps. This is the story right now. I'm just trying to work. So at the end, connecting natural history, heritage, and habitat, I mean, very important for the future conservation. So integrating genetic, ecological, and cultural insights reveals evolutionary stories and calls us to protect the diversity that shapes our natural and cultural heritage. I mean, this is my story. Thank you very much for your patience. I hope I could explain, but I tried. Thanks a lot.
I spent a little time in Africa, and I did see one Turacos high up in the trees. Have you done any research in the field, or is it all museum-based?
So this is, I mean, I didn't mention, this study mostly based on the museum specimens, and I think that makes this study a little special for me because I didn't go to Africa, I didn't see the Turacos in the real habitat. But I tried to use all these museum specimens to get the DNA, and I believe the museums very important to understand the biodiversity. I mean, all these historical collections, I mean, invaluable. I mean, yeah. It's very, very important.
Well, thank you.
Have you tried reaching out through organisations like the African Bird Club to try and...
Yes, I did. Okay. So they, for Heart Lobby, we tried to go to Kenya to get the recordings. But I was working in the American Museum. I tried to get some grant from the American Museum. They can give the grant, but there's a tradition there. If you go Kenya and collect birds, you can get the grants. But if you go to Kenya and collect songs, they are not going to give you the money. So, so the collecting, I mean, it's very important for America. So there's a kind of different tradition there. So, and I couldn't get the money, and I applied to different funds because I arranged everything for the field work, go to Kilimanjaro, go to Usambara, try to get these recordings. But I couldn't get enough money. But a couple of months ago, something, I mean, appeared. So, I mean, one organization in Turkey, I only needed just $10,000 to go there, to do this field work, get recordings. And yeah, I found somebody. I just mentioned this study, but I didn't get any intention to get any money from them. I just explained my study. I said I wanted to go Kenya, and then they said they wanted to, support me. So I said, that's fine. And it might happen the next spring to go there, to get some recording, to clear up the situation for the heart lobby. Yeah, I just tried to do something, but I didn't get any success before.
Do you think that the recording of and greater understanding of calls is going to be key to unraveling some of all this?
We try to use some xenocounter recordings, but they are very limited.
Yeah.
And we recognize there might be difference, but it's not a very good quality. And there is no statistics under the scene. So we only hear it. I think it's go there and it's worth it to do that. But I don't know. It's not really for sure.
Does what you're saying mean that you should be grateful to all the previous
Yes.
generations of people who were interested in birds, that
Yeah.
they went around shooting them, so that there are all these specimens in museums for you to have a look at? Because now, presumably, you can't get away with shooting these things in any country.
Yeah, that's why we have to protect all these historical collections. Right now, it's not really worth to kill the birds to collect them, but we can use the historical collections to understand the biodiversity, and we can make some solution to today's biodiversity crisis, because the planet's main problem right now, the biodiversity crisis, not a climate change, not a global warming. So if we find any solution for the biodiversity crisis, this is the main problem. The climate change looks like a symptom. So we are going to lose climate change automatically. But we are making deforestation, we are making habitat destruction, we are making pollution, everything. I mean, we really affect all these creatures, all these species, then we are getting these climate change, global warming problems. So people solving the global warming problems, it's correct. It's definitely, I support. But I think nobody talks about the biodiversity crisis enough. So if we solve this biodiversity crisis, we are going to automatically solve the global warming problems. So that's why all these historical collections, I think, very important. We have to use them in a proper way.
You mentioned your ethnographic work with Andy Gosler.
Yeah.
What sort of publication? That's ongoing, if I understand correctly.
Yeah, just started, yeah.
What sort of publications do you see coming out of this?
After I finish this work, it's going to be the huge work, but we are going to pick up at the beginning, we are going to pick up some representative groups. It's going to be, probably some research article is going to be coming up. So try to understand the bird names and losing the bird names, some cultural information, biocultural information. We try to make some connection, the culture and the biodiversity. And I think we can use this information to make some publication like Journal of Ethno-Ornithology or Journal of Ethnobiology or something like that.
Right, so you're not necessarily looking for a monographic type of publication, but on
No.
various papers.
Yeah, definitely. Yeah, definitely.
Because you're based at Tring at the moment, in
Yes.
terms of using the collections.
Right.
I think you're with us for 2 months.
Yeah, the idea is end of the December,
Yeah.
but if I need any extension, so I'll definitely tell you and Alex and the other people. So, but I think I try to finish within 2 months all these museum work, then move to Oxford to writing and analyzing stuff.
So at the moment, you are databasing extracting information from individual skins,
Right,
roughly how many Turacos are in the Natural History Museum? I don't know. But
800.
yeah.
In the Tring collection. Yeah, with grey go-away birds, more than not 900, between 900 and 800, I remember. Yeah, definitely. Yeah, I measured all of them. It's a huge numbers. And also in American Museum, the same amount of Turacos also over there. Well, not same, probably between 400 and 500.
Yeah.
So between two institutions, actually, we have more than 1,000 Turacos. I mean, it's very important. And if you go to the Field Museum, they have really nice collection from Africa, another probably 500 Turacos over there.
And you haven't started looking at the eggs yet?
No, I have no idea about eggs. Yeah.
Right. Thank you, Utku. That was lovely.
Thank you very much. Thanks a lot.